In this video, I'll present a short demonstration of ImageBreed, which is a completely open source system for managing agricultural field experiments. The same system can be used to manage greenhouses and other controlled environment experiments as well. So we use unoccupied aerial vehicles to fly over our field experiments, and we capture a series of images as we fly. However, what we ultimately want are analytic results, such as which germplasm is performing optimally, or information about any field variation, such as soil heterogeneity or other effects in the field. The first step in the image processing pipeline is to generate an orthophotomosaic. You can choose to use external software such as Pix4D or Agisoft. However, you can also simply upload the raw images to ImageBreed, which will then use OpenDroneMap to create these ortho products and then save them directly in ImageBreed. This can save you time and can produce more standardized results. OpenDroneMap is an open source software for creating these products and can work for both color images and for MicaSense 5-channel multispectral images. Afterward, you can then use simple interactive web tools for assigning the plot polygons onto many different imaging events over the growing season. Here you see many imaging events on this field experiment across the entire growing season. You can use ground control points to standardize the templating and can double check that the templating is accurate. After you have the plot polygons defined, ImageBreed will then extract phenotypic values for each plot, such as vegetation indices and other auto-encoded computer vision type traits. However, there's a very large amount of metadata associated with what I just showed you, such as what germplasm is planted in which plot in that field experiment. ImageBreed is a web database for managing the germplasm. For instance, here are different maize hybrids and different barley sessions. You can also associate complex pedigrees onto these germplasm to track selections throughout time. You can have many field experiments in the system. Here's the interface for viewing different field experiments grouped into breeding programs and arbitrary folder structures, which you can define to help you organize your different field experiments. Each field experiment also tracks the physical layout of your experiment, where each cell here represents one plot where you planted a specific germplasm and its spatial position. ImageBreed also stores location information for these field experiments, so you can have large multi-environment trials, as you can see here. Furthermore, you can have metadata for each accession, such as genotypic information for that germplasm. For instance, you can upload a variant call format file defining very large numbers of genotypes for each germplasm. Here showing close to 1 million genotypic markers for this genotyping protocol. And then using that information, we can finally automate our analytics. For instance, combining multi-environment information to determine which hybrid is performing best. And I'll have a number of analyses currently in development. I'll be uploading many other short videos such as this in the future, so stay tuned. I'll describe key aspects of the program. And please follow the links provided to learn more about ImageBreed.